All right, today we are opening a cold case file that is literally billions of years old. It's a fundamental mystery right at the very heart of biology. We're investigating the case of the one-handed molecule.
And it all boils down to this one single question. Why, out of all the possibilities in the universe, does life on Earth exclusively use one hand when two are available? I mean, think about it. That's a profound puzzle, and the answer has huge implications for how life even got started.
So what do we even mean by handed? Well, just look at your own hands. They're perfect mirror images of each other, right? But you can't actually stack one perfectly on top of the other. You can't fit a left glove on a right hand. It just doesn't work. Weirdly enough, molecules can have this exact same property, and we call it chirality. So you have left-handed, or L molecules, and right-handed, or D molecules. Chemically, they're identical, but their structure, total opposites.
Okay, Section 1. Life's one-handed mystery.
So why does any of this matter? Because this molecular handedness isn't just some quirky little detail. It is a fundamental organizing principle for all life that we know of. When you look at the universal features of life, from cells to metabolism to genetics, one property just screams out at you: homochirality, and that just means same-handedness. The amino acids that build our proteins, they are all left-handed. The sugars that form the backbone of our DNA and RNA, you guessed it, they are all right-handed. And there are almost no exceptions to this rule. It's kind of nuts.
And this is not a coincidence. It is an absolute, non-negotiable requirement for life. Without this uniformity, you just can't form the stable, predictable structures that life depends on, like that elegant spiral staircase shape in proteins called the alpha helix. You throw just a single right-handed amino acid into that chain, and bam, the whole structure just breaks. Life needs order, and homochirality is what provides it.
Section 2, cosmic forces or chance. So, how did this incredible uniformity happen? For a long time, the investigation had two main suspects in the lineup. The first is just pure chance, like a random fluke of crystallization. The second is cosmic forces, like some kind of special polarized starlight. But you know what? Both of these ideas have a major flaw.
See, the problem with chance is all about scale. Sure, you might get a pond over here where, just by luck, left-handed molecules happen to crystallize out. You get a nice little local pocket of homochirality. But a few miles away, another pond could just as easily favor the right-handed versions. On a planetary scale, it all just averages out to 50-50. Chance just can't explain a global, consistent bias.
Okay, so what about the cosmic forces idea? Well, that one's even trickier. The leading theory is that a certain kind of polarized light from space could create a small bias by, get this, destroying one molecular hand more than the other. But the effect is tiny. We're talking maybe a few %, and it's a destructive process. Even worse, it's not a one-size-fits-all solution. For some amino acids, that same light would actually favor the opposite hand, completely breaking the uniformity we're trying to explain. So that theory's got some serious holes.
Section 3, peptides, then everything else.
With our classic suspects looking pretty shaky, the whole investigation took this massive turn, right into a classic chicken or the egg problem. See, life uses homochiral enzymes, which are proteins, to build other homochiral molecules, like the sugars in our DNA. But how did you get the first homochiral enzymes without already having a system in place to build them? It feels like a total paradox.
But a really compelling idea from a researcher named Soren Toxford suggests a way out of this paradox. He proposes a very specific sequence of events. First, you get homochiral peptides. These are just short chains of amino acids. Only then, using those peptides as your first tools, do you establish metabolism and genetics, which eventually leads to the first cells. The order matters.
So the crucial bit here is how those first peptides got their act together. And the answer is amazing. It's not about chemistry, it's about physics. When left-handed amino acids link up, they naturally fold into this really stable compact helix shape. This physical stability made it way more likely for all L chains to form. Sugars, on the other hand, don't have that advantage. They became homochiral later because those first L peptides acted like primitive little enzymes selectively building only D sugars. So peptides basically solved their own problem first, and then they went on to solve the problem for everybody else.
And get this, here's a totally wild twist. The exact same chemical property that makes amino acids unstable enough to sort themselves into those perfect helices, well, it's also our Achilles heel. Over a lifetime, our L-amino acids can spontaneously flip back to their D form. This process is called racemization. It damages our proteins and is now being linked to aging and diseases like Alzheimer's and cataracts. The very thing that allowed life to start might also be the secret to why it ends.
Section 4. A violent beginning.
But this still leaves one huge question. Where did that very first little bit of bias come from in the first place? A radical new idea called absolute asymmetric synthesis suggests it wasn't selected from a 50-50 mix at all. Instead, it was forged with an inherent bias from the very beginning, a bias that's rooted in the fundamental asymmetry of the universe itself. To test this wild idea, scientists designed this absolutely incredible experiment. They used a gas gun to fire projectiles at over 7 kilometers per second to simulate a meteorite impact on the early Earth. This insane collision created something called a plasma torch, a superheated cloud of atoms. And inside that cooling plasma, amino acids were actually created from the simplest ingredients.
And here it is, the smoking gun. When they analyzed the amino acids that were made in that plasma, they found a consistent excess of the left-handed form, somewhere between 7 and 25%. Now that is not some tiny destructive little nudge from starlight. This is a significant, constructive bias created in a violent flash. It suggests the building blocks of life, whether they arrived on meteorites or forged in impacts here, were already fundamentally skewed to the left.
Okay, Section 5. A universal blueprint?
So where does all of this leave our case? Well, we've seen that the old suspects, chance and distant cosmic forces, probably weren't the main culprits. The evidence now points to a 1-2 punch. First, a violent beginning. Huge impacts forging amino acids with a significant built-in L-handed bias. Then, here on Earth, the unique physics of peptides took that little head start and amplified it all the way to 100% purity, setting the stage for metabolism and genetics to take over.
And that leaves us with one last, truly mind-bending question. If life's handedness is forged by the fundamental laws of physics during these violent cosmic events, does that mean that any life, anywhere in the universe would be built from the exact same L-amino acid D-sugar blueprint? Are all proteins everywhere left-handed? Are all genetic codes right-handed? The case might be closed here on Earth, but it looks like it's just been blown wide open for the entire cosmos.